Hello, my name is Cheryl Belanger, and the topic I am covering today is neighboring weeds influence the formation of arbuscular mycorrhiza in grapevine. The abstract of this is that grapevine, Vitis vinifera, ribwort plantain, Plantigo lanceolata, and Dalmatian pyrethium, tenacidum cineifolium were examined in pot culture experiments, individually or when combined, to see whether multiple hosts influence the formation of the symbiosis with arbuscular mycorrhizal fungi, also known as AMF. Vitis vinifera and T. cineifolium, individually and in combination, resulted in a greater development of arbuscular mycorrhizae in terms of spore production, extra radical mycelium length, and root colonization compared with those containing P. lanceolata. Herbaceous weed species seem to promote a different set of dominant AMF, potentially providing a wider spectrum of AMF for colonizing grapevine roots. This indicates the value of encouraging host plant diversity in vineyards. The introduction states that the establishment and maintenance of the plant community and ecosystem processes are greatly influenced by the associations between plant hosts and arbuscular mycorrhiza fungi. This association is commonly referred to as a symbiotic relationship, inferring that the composition and diversity of the plant community can influence the structure of the AMF community. Thus, apart from environmental factors, AMF diversity is dependent on plant species, identity, and age. As well as plant host specificity, AMF composition is influenced by neighboring plants that can have beneficial, inhibitory, or neutral effects. Plant competition as natural ecosystems may be indirect and depend on the establishment and maintenance of mycorrhizal networks and the nutrient exchange within them. Vitis vinifera is a crop whose establishment and growth highly depend on the presence of AMF. Grapevines have a low root density compared to other crops, and its AMF symbionts play a significant role in its water and mineral acquisition. The formation of grapevine mycorrhizal networks with neighboring plants depends on vineyard soil management practices. The way that this was carried out through methods and materials can be seen as the plant material as the hosts Vitis vinifera, Plantigo lanceolata, Tanacetum cineifolium, Taraxium officinale, and Trifolium repens. Grapevine cuttings collected from the vineyard near a town in Croatia were disinfected and then dipped into an IAA for root growth promotion and planted. Weed seeds disinfected and washed with sterile water were germinated with the grapevine cuttings. 3 weeks later, the seedlings and rooted cuttings were transplanted to 2-liter pots and fertilized with a 15-9-15 NPK fertilizer. Inoculum was carried out by grapevine soils that were collected from the 4 sampling points in the vineyard at bud break stage. The sampling points were 10 meters across the diagonal of the rectangular vineyard. The soil was collected at a depth of 0 to 20 centimeters. The samples were mixed to make a sample of 10,000 grams. In each 2-liter pot, 100 grams of mix was added to the pot below the seedling's roots. The pH of the resulting plant growth substrate was 8.0. After the experiment was conducted, the results are as follows. Prior to planting, host seedlings and rooted cuttings, spore abundance was 5 spores per gram. After the inoculum was added, the estimated spore abundance was 0.26 spores per gram. 2 months after, the combination of GR and TC exhibited the greatest abundance of AMF spores. After 4 and 6 months, it confirmed that GR-TC and GR plus TC treatments as the most suitable for spore production, with maximum values being about 6 spores per gram. Average fresh and dry shoot weight, root fresh weight, and fresh shoot root ratios are shown in Table 1. The greatest shoot biomass was measured in grapevines, while the highest root shoot ratio was calculated for Plantago lanceolata. The fungal 18S rDNA sequences obtained from the host plants were submitted to the GenBank, and 43 out of the 80 sequence clones belong to Glomeromycotin. The cluster analysis of distribution of AMF among different plant hosts showed the highest similarity between AMF communities of T. officinale and grapevine. The AMF community on the roots of P. lanceolata was the least likely AMF community of other host plants examined. To further discuss this, the results confirm the initial hypothesis that the host plant identity and density affect the development and composition of the AMF community. The grapevine and the T. cinerifolium, individually and in combination, were very supportive in terms of mycorrhizal establishment, and were a significant improvement compared with P. lanceolata and its combination with grapevine. The findings indicate that grapevine does not always have the same facility to interact with AMF, which is dependent on its neighboring plants. Plant root exudates may be involved in suppression or stimulation of AMF root colonization. The influence of root oleochemicals was explored in P. lanceolata as a highly mycorrhizal plant. The root exudation of P. lanceolata does not reduce the potential benefit of symbiosis with AMF, although it serves as a defense mechanism. Grapevine, T. cinerifolium, and P. lanceolata developed great shoot biomass when planted alone rather than in higher density or with other species. This confirms the disappearance of the positive effects of AMF on plant growth when plants are more crowded. In conclusion, not all plant interactions are equally favorable for mycorrhizal establishment. In these relationships, the essential and specific characteristics of each plant species are important, and diverse allelopathic effects may occur. P. lanceolata, when a neighbor, reduced the development of grapevine mycorrhizae. In contrast, T. cinerifolium was a beneficial grapevine neighbor from the aspect of AMF development. And in the bottom corner is 2 photos of the first being T. cinerifolium, and the second is a photo of P. lanceolata. Thank you so much,